Hello, everybody. If you're here for Science and Nature, you are at the right place. I'll just leave a few seconds for people to either log into the Zoom or, catch us on the Facebook Live, and then I'll begin. Okay, so I think that was a few seconds, so I'll get us started. So welcome back to Science and Nature Untapped. Again, we'll just do a little bit of housekeeping before we begin. And so this is for everyone who's watching live on Facebook or on Zoom. We always love to know where people are tuning in from. It's nice to see kind of like the range of viewers. You may add your name if you're on the Zoom. On Facebook, we might automatically already see it. And then if you have any questions for our presenter, which we'll get to at the end of his talk. So I'm just going to do a little countdown from Gabe at your TV. So 3, 2, 1. Gabe, it's all yours. Hello, everybody, and welcome back to Science and Nature Untapped, a free virtual monthly speaker series brought to you by the River Institute. My name is Stephanie. I will be your host for this evening. Before we begin, we'd like to acknowledge that many of us are living on unceded land and traditional territory of Indigenous peoples. We are grateful for the opportunity to live where we do, and we thank all the generations of people who have continued their responsibilities to Mother Earth since time immemorial. In case you're not, basically, we don't say specifically whose land we're on, since this is live and we have people tuning in from everywhere. We are on Haudenosaunee land if you're living in Cornwall and along the St. Lawrence River. But if you're elsewhere, I highly suggest that you take a look at the website native-land.ca, and then you'll get a map of the world, and you'll be able to see whose land you're on, whether it be where you're traveling to or where you're living. So native-land.ca. If you'd like to learn more about what we do at the River Institute, the projects, our research, our education programs, who we are, you can follow us on Twitter, Instagram, or Facebook. In case you'd like to see any of our previous talks for Science and Nature Untapped, they're all recorded and can be viewed at riverinstitute.ca/ontap. So for upcoming events, we have a series of workshops that we put together as part of the Great River Rapport project. All of these are on the River Rapport website at riverrapport.ca/workshops. We're continuously updating that page. So if you'd like to have updates, you can subscribe to our mailing list. Our upcoming event will be with Dr. Marianne Perron, who will be helping you identify dragonflies from your photos. This should be an amazing opportunity to speak to a dragonfly specialist. And so we hope to see you there. If you do have some photos and you'd like her to take a look at them before the presentation, you can send your photos to maperron@riverinstitute.ca. So that's maperron@riverinstitute.ca. So a lot of the research that we do, education programs, projects, are generously sponsored by donors. If ever you would like to support us through a donation, you could go to riverinstitute.ca/donate. But as I've said before, just speaking to your friends, to your family about us, or sharing on social media goes a really long way, and we appreciate everything that you're doing. So thank you. And so for tonight's talk, we're going to be hearing about water-fish community, challenges and opportunities for post-pandemic science with Dr. Barry Madison. He takes an integrative molecular to population level approach to his research. He draws on his broad research experience employing techniques from physiology, endocrinology, and toxicology to study animal responses to environment and climate change. In this presentation, he will provide some perspective on his background and approach to research, as well as the story of why water, fish, and community represents a new age of integration in his scientific journey. Recent events have caused many scientists to reflect on what is important to them, and he hopes to demonstrate how COVID has made many scientists rethink their approach to research. So thank you so much, Barry, for taking the time to be here with us tonight. And without further ado, it's all yours. You may share your screen.
Thank you very much, Stephanie. Just going to share my screen here. Should be up. Is that good? Can everybody see it? Okay.
Yeah, that works.
Okay, thank you. So thank you everybody for joining me tonight, and thank you for inviting me to this special series. A lot of this has to do with, what I thought would be an interesting story about, my work in the area for almost 20 years now down here, even though I may not look at my beard, definitely does. And basically, it's going to cover three major themes of my research and my experience: water, fish, and community. Now, the community really has to do with the Cornwall region and the local area here post-COVID. So it's the first half of the talk is really going to be talking about my research experience and what I've been doing for many years around here. And then basically what's happened since COVID and how we've kind of, as scientists, been forced to kind of rethink how we do our work and to reprioritize our work as well. So thank you for joining me in this. So the subtitle of this talk is going to be Challenges and Opportunities, because it's not just the challenges that we run into. We actually found a whole bunch of really cool opportunities in post-pandemic science as well. So just to highlight a couple of things to show off in my background, I'm from a very small town up north. It is known as Kenora, Ontario. It is up in this region here, and I got very lucky to grow up in that area, very privileged to be in Lake of the Woods, and I'm actually in Keewatin is where I'm from. But basically, I had access to water and fish and a very important community. I'll come up later on at a very early age. And, basically when all my friends were out having a good time partying, I was out on the water looking for fish. Now, these are all fairly recent pictures within the last decade or so. It's because I started doing all this before the advent of digital cameras. So I'll have to go digging through a lot of my old photos to find this stuff. But this got me interested in water and fish, extremely important to me at a very early age. I got almost obsessed with it to the point where I think I figured out the stressing part of fish fairly well by fishing for them all the time. I was a catch and release angler. As you can see, many of the species here will be around native to the St. Lawrence River as well. But basically, I decided to do something about that after I learned that I was just stressing fish, and maybe I should learn a bit more about the actual physiological mechanisms behind what stress actually is. And so I got really lucky again to join Queen's University. Now, this will be kind of the focus of how I run through my talk in terms of format. The left will show the content or the theme of my research, the particular degree in the background, and then who I was working with at the time. So some of these names may be familiar with you, to the audience here, and they're at Queen's. They're Yishang Wang and Dr. Bruce Tufts. And basically, I got really lucky early on to join a project that looked at one of my favorite fishes, walleye. And as an angler in Kenora, coming down to Queens down here in Kingston, I was very excited to work on a species that I thought I knew a lot about. It turns out they're quite complex, as most fishes are. But this is why I call this whole chat, this part of the talk, the walleye years, because basically what I was charged to do is to look at novel methods of potentially culturing walleye for stocking purposes or for just, alleviating some of the fishing pressures, because probably recognize how important of a species this is for local fisheries. Specifically, we had some challenges associated with our culture with nitrate and ammonia. So at a very early on in my career in research, I started understanding how water was important and the things that are in water are important and how they interact within animals. And particularly, I was interested in how stress and growth worked. Now, this led to my very first paper many years ago, and it basically is still one of my most cited papers, not because it's the oldest, because somehow there's a following of people that want to look at nitrate exposure with walleye. But basically what there'll be some data in this talk or some data moving forward, but don't worry too much about the details. If you've got questions for you, I have no problems elaborating on some of these. And there's also available to find some of these older papers as well. But basically in looking some of the background research here, we saw that, something like ammonia, for example, that is typically considered really bad to fish, especially in water. It very easily dissolves in water and it's very neurotoxic. But it's also a source of nitrogen. And what we saw basically in a series of experiments that ammonia can actually and nitrate as well can actually have a physiological impact on fishes that make them a little bit more of a challenge to culture. And for example, walleye here, we found that the oxygen dynamics and how they responded to changes in nitrate really made them a challenge to culture. As most of you know, maybe that they do flow or they do move from deeper to shallow areas to hunt. One of the cool things that they also do is they're very voracious eaters. And if you put one of them in a tank as a juvenile, you'll end up with one of them left after one has eaten the entire cohort of fish. They're very interesting fish, but they're very difficult to work with in a culture scenario. So the hot button ticket was, how do you get these things to grow in tanks and then put them into the water essentially for stocking purposes? It turns out that you basically need dirty water, because if they can see each other, they will eat each other. And it's one of these really neat scenarios with, you didn't expect to see that when you first started doing the research, but that's how you have to argue with, your animal care protocols, for example. But overall, what we found in this particular, in this work, was that managing amounts of ammonia is as important as removing it, if not more so. Meaning, if you're removing ammonia from a system, you're actually removing nitrogen that can be converted into muscle. And if you're converting muscle, that means you're making mass, and mass is fish muscle, which is what you want to eat. So basically what we have here is a sink of nitrogen. So if you can manage ammonia and build up in the animal, you're actually getting more bang for your buck by making them larger over time. So it's a really interesting theory. It also has, it's had its challenges out there, but basically it was eventually vindicated when we found out that we can radio label ammonia that comes out of the animal in aquaculture, or sorry, we can stable isotope label, I should say. And then we can see that the fish actually reincorporates that ammonia back into its, that nitrogen back into its body afterwards. So it's a pretty cool effect. I then got really interested in what this whole stress thing actually means. And I found out, digging into the literature, that basically the stress itself, the word stress, is nothing more than a challenge to homeostasis. I mean, this is really because any set point that an animal is trying to maintain is their ideal conditions in that particular environment. Any change to that requires a series of responses to respond physiologically within that animal to those changes. We know a lot of these things on the left-hand side of the screen here to be our typical things we consider stressors. We know things like pollutants are stressors, low oxygen is a stressor in fishes. We also know things like the physical stressors, like when I was out fishing for them all the time, confinement, transport. These are all big stressors to fishes. But we also know that the perceived stressors can be as important as the stress, at least at a physiological level. And to me, this was quite surprising. So what I basically had done by the end of my master's is figured out, well, I really like this stress stuff, but I don't really know much about what happens downstream. And you can see all these other things, these primary responses, these secondary responses, and tertiary responses. These are a series of physiological stress mechanisms that occur essentially once something is actually stressed in the fish in this case. So a primary response is to your typical increase in glucocorticoids or cortisol, these steroid hormones that help liberate energy. And that liberated energy helps the secondary response essentially prepare the body or that animal to get out of there. And that is a fight or flight response, if you will. When you get a large enough or a long enough or a chronic stress in this case, you can start to see responses that are changes that show changes in tertiary aspects of the animal. So you start to see changes in how it grows. You start to see how it behavior is modified and how it swims and things like that. So I started to understand I need to know more about what's actually going on here. So this is from moving from the animal physiology section. I decided to enlist some efforts of a very well-known neuroendocrinologist, a comparative endocrinologist, University of Guelph, and his name was Nick Bernier. And what he taught me were many things, but basically we spent several years doing some pretty intense endocrinology work in an area that I call the last zoologist, or the seawater phase. I don't say this to toot my own horn. It's basically the fact that I was at University of Guelph when the Department of Zoology became the Department of Integrative Biology. And they had asked me if I wanted to keep that degree, integrated biology or zoology. I chose zoology. So I'm the last zoologist from from University of Guelph. But basically what had happened to this is we needed to study how the actual stress mechanisms control things like growth and feeding behavior and appetite and all those other responses that we were looking for in fishes. And what we ended up doing is a series of really intense experiments, but there's one really cool one we did, and I suggest you check it out, is essentially we maintained an artificial state of high cortisol levels in a fish representative of a chronic stressor. Now, typically, cortisol is released in a pulsatile fashion because it has rhythmic-based physiological responses that the animal needs in a day-to-day operation. So they're not typically, this hormone isn't typically elevated in high levels and maintained at high levels. Now, when I say high levels, they're fairly low levels overall, but it's such a very metabolically active hormone that very small amounts of it cause very large changes. Anyone who has undergone stress knows that cortisol is a pretty nasty thing, especially for humans and their ability to digest fat as a result of that. But in this case, we were looking at how we get a response from stress in fish without having a typical cyclical manner of cortisol. If we just elevated it artificially and maintained it there, what would happen? And we see all sorts of stuff happen within the fish, from it literally catabolizing its internal stores in sequence. And so at the end of this scenario, you have, say, 28 days or so of this fish that's been sitting with an osmotic pump that delivers cortisol into its body. And it looks very emaciated and very sick at the end of it, almost like we would expect to see. But what this allows us to do is to tease apart the specific mechanisms within the brain and the link to the tissues that drive metabolism to show how stress actually works in fish. It was a really important paper. Just really cool stuff that had happened. We also looked at basically whether or not the internal generation of cortisol within the fish was affected. Basically, if you artificially create a scenario where there's a lot of cortisol in the body, the internal mechanism tries to shut it down, but it can't. And so we artificially looked at what happens when you really stress a fish out. So this is a pretty cool paper that showed, a very comprehensive view from start to finish, essentially, what happens over 28 days of dosing with straight cortisol and then a whole recovery period after that. We also looked into some aspects of how this affects the immune system. And so we really wanted to know how stress affects growth in fish, as well as just stresses, or sorry, parasites affect growth in fishes. And so basically all we see here is we see this neat little this natural parasite called Cryptobia salmacitica. It's very prevalent in the West Coast and salmonids. And what it actually does is it gets into the bloodstream, and then you can see the next picture here coming up is a red blood cell. And this actually will attach to the red blood cell and slowly, liquefy it and eat that, if you will, consuming that as energy. And so you get these really nasty looking effects, these popeye and these enlarged spleen as a result of this disease. But we also see that it reduces the ability of the fish to generate that stress response. And this is just exemplified by showing things like when we spike their actual kidney tissue or their adrenal tissue with these precursors of cortisol, you'll actually see them still be able to respond, but it's at a much more of a muted level because they're just sick. And so this parasite has a really unusual component of it. It's instead of letting cortisol liberate energy that it can then use to grow and propagate, it actually mutes that response. So we thought that was a pretty cool aspect of that. And we're still working on that mechanism as of today, actually. So one last component of my PhD is that we also did a whole bunch of environmental and genetic work. Now, we really wanted to understand how the stress response is affected by both lineage, or parentage in this case, so parents, as well as rearing environment or early rearing environment. And so what we had done, and I worked out west in a really awesome place called Yellow Island Aquaculture. It is a Chinook salmon farm, organic Chinook salmon farm in Campbell River. Well, it used to be in Campbell River. It's in Quadra Island. And the owners there are really interested in research, and they really facilitate a lot of research collaboratively with a lot of different people, which is really cool. But one of the things we did is we used their brood stock that they had for, I think, 20 or so years from the natural populations in the area. And we started to manipulate the environments in which the fish were being reared. So within their own brood stock, we would take them and either artificially rear them like you normally would by just mixing random Milton eggs together, or by letting them choose their own mates in a artificial channel, and then taking the actual eggs from that and then rearing those fish up to see if there's a difference of allowing the fish to choose their own mates. We also did something that's in between that allows the fish to choose their own mates, have the eggs, take those eggs out of the reds or the gravel-based, the eggs are sitting in the gravel, and then rear them in an artificial environment. So we physically got the genetic component, but the environment's different in which they're being reared. And so we end up doing a lot of experiments on this lineage system. And that cover everything from their growth to their seawater transfer ability to behavior to aggression and things like that. And basically, it was this really cool scenario where we were able to mix and match lineages and environments. And it's pretty cool because basically what it turns out is the type of environment to what happens in a hatchery, a very intense growing scenario, very competitive, leads to these very different behaviors, but also leads to very different fish at the end of it. So much so that it actually a lot of the times overrides a lot of the genetic components, or at least the genetic advantages to begin with. Now, remember, this is all in a hatchery. So once these fish reach the seawater phase and grow up into full-size fish, I mean, that's a larger study. But at this point, we saw some changes that we could say for sure rearing environment and the actual lineage or parentage have large implications on how much a stress or how much a fish responds to stress. This led out a bunch of papers, and as I said, they did cover a bunch of different things that from either physiological performance and growth to even things like behavior. Where so much so that even a colleague of mine, Sean Garner, had found some evidence of salmon eating each other's eggs in the streams, which had not been shown previously, or at least was contested. So just some of these random cool things that are associated with doing fish work in their natural environment. So because of all that, I got really interested in comparative endocrinology, but it's very lab-based stuff. It's very, very involved. There's a lot of protocols to do, a lot of clean work, a lot of back and forth. And I realized, you know what? There's also a really important component that I'm missing here that's really relevant, especially these days, and that is contaminants. So I decided to leave Guelph, come back to Queen's to do well, it's actually through Queen's and Royal Military College. So I got an opportunity to come back and work in an ecotoxicology lab with Dr. Langlois at RMC, as well as Peter Hodson. I'm sure many of you know, you may be listening, at Queen's. And so basically, I call this journey the wading into petrochemicals. Now, this is an interesting journey because I came fairly green to it. Again, I had just got off of being in my PhD and studying lab-based stuff in neuroendocrinology, and I'm walking into petrochemicals. The reason why I say that is because petrochemicals is a fairly hot button issue. And it was at the time where the oil sands were just everywhere in the news kind of thing. So dilbit or diluted bitumen, which are the main products coming out of the oil sands, was novel. It still is fairly novel in terms of it's a mixture format, but we didn't know much about it. We didn't know much about its toxicity. We didn't know what would happen if say a pipeline burst somewhere along the way that was carrying dilbit. I mean, we knew that from 2010 in Kalamazoo that some pretty major issues happened as a result of this. And this is because this particular compound itself more or less represents asphalt in its native form. It's oil sand. So a bunch of stuff have to be added to it to make it pipeable or pumpable, if you will. And these are just oil gas condensates, essentially, that make something that's this brick or bitumen-like form to this conventional oil type density. And so basically those components are diluents, and they're added downstream somewhere, or upstream, I should say, somewhere, and then downstream into a pipeline. So what you get is a product that resembles a brick of bitumen at the beginning of it. You add this diluent to it, you can move it somewhere, and then if that evaporates on the other end of it, you get a brick again. And so basically we know from what happened in Kalamazoo River in 2010 is that some of the challenges with this particular compound is once this oil weathers in the environment, you get a very sticky tar-like substance that coats the bottom of the river in this case. So, how we work with this stuff, it was novel. We didn't necessarily know how it was toxic or why it was different than other conventional oils. And a lot of this had to do with the fact that this was a very novel mixture. It would vary from year to year. It would vary from season to season, or who made it. So all those other components, those proprietary components, the diluents, etc., we didn't necessarily know what was in them or how toxic those components were in addition to the bitumen itself. And so what we end up doing is making these kind of exposures using fishes, developing fishes. And we're using things called WAFs. These are water accommodated fractions. And what these are essentially just mixing oil and water. We've all been taught more or less when we were younger that oil and water don't mix, but there are components or soluble components of oils that do. And these are PAHs that get into this that cause all sorts of stuff, issues in fish. And so our WAF, for example, is we just take oil and water, mix them together with some energy, and then we use the water underneath the oil and with no oil on it, expose fish to that and to see what happens. Another version we use is called a CWAF, which is a chemically enhanced version of a WAF. And all that is adding dispersant. These two scenarios more or less represent your oil spill and almost your cleanup scenario. Now, I should say this with a caveat because you don't normally add dispersants in a low energy system like a river or a lake that's fairly small, because there are toxic components in those dispersants as well. Typically, dispersants are used for larger energy systems like in the ocean, because the mixing energy of the ocean, wind, water, wave action, allows it to disperse into smaller micro droplets that then get degraded by bacteria. And so in this case, we were interested in just using dispersant to get more of these soluble PAHs into solution, because that's its job. So we use these two different versions of these WAFs, if you will, to test how toxic they were to a bunch of different fishes. Now we have to start where we knew most things. Now there's a bunch of different animals across animal physiology that are used almost like your white lab rat consistently. And one of them happens to be Japanese Medaka in fishes. And basically it's because of the way their developmental period and how much information we know about them. And in this case, we exposed these fishes at a particular time when they're developing. So right after they become, it shows right here, right after they become an eyed egg, essentially, you put them in these exposures and you get these long-term chronic solutions that you expose the fish to. At the end, these fish hatch with malformations, and the malformations they're then ascertained as to how severe they are, and we can actually build exposure-based metrics on what they look like after the result of this. On top of that, we also then take a bunch of them and grind them up and look at their molecular signals within fish to see if we can ascertain what's actually happening at a molecular level to some of these fish during these exposures. Now, when you see a component like this, all this is showing you essentially is this percent affected is just showing you how much of the fish are malformed as a result of their exposure. On the bottom here, you basically see what we're looking for in the oils. This happens to be the total PAHs that we're looking at that we know cause these type of tumors and effects and things like that. And what we're showing is how the different concentrations were differed between WAFs and CWAFs. You can see that basically that CWAF with that dispersant gets a lot more of that stuff into solution. So we were basically looking to see what kind of molecular effects we can see in fish that are specifically related to this oil's exposure response, that if we have to look across different mixtures of oils, how can we compare these things, right? How can we compare apples to apples, as it were? And one of the interesting things we found out is one of the very high-level enzymes that respond to xenobiotics is CYP1A. And we found that CYP1A actually responds very well to oil-based exposures, almost such that you can get a dilution series that almost represents exactly what the concentrations that you're doing. And so just to see these immediate responses in fishes, we were like, okay, well, we see what happens when we have these very malformed fish. In fact, I've got an example of one. Here's one right here. What happens after a fish has been exposed to oil for a while is something that we see here called, well, they're malformations, but in a group we'll call blue sac disease. And these are a combination of different types of malformations. TH in this case is referring to a tube heart, and a PE is referring to a pericardial edema. Now, the edema is nothing more than the fluid filling up in the pericardial region, causing the sac around the heart to be essentially filled with fluid. What it has a secondary function is stretching that heart very far apart from where it normally would be, giving it the effect of a tube heart. You can tell, I'll show you this video right here, that this would have an implication on the ability of that animal to circulate blood properly when it's developing. If you can't circulate blood properly, you can't fuse or perfuse certain tissues properly, you can't grow them properly, you get malformations. So these are how we figure out what happens in our exposures with our eyes. However, we also did this across a variety of local species to see if our model species act similar to our local species. We know this because as fish physiologists in our training from before, we know that a lot of fishes have a lot of different cool adaptations. And those adaptations can really leverage or dissuade you from using them in certain tox tests. For example, and just there's some of them down here, the ones that are highlighted here, Japanese Medaka and rainbow trout, happen to be very popular models of fishes that we use for our research. But they're also very specialized fishes. For example, the rainbow trout here is a cold water fish that's very sensitive to oxygen stress. So if you're doing a component of where you're not sure what the oxygen is in your exposures, you could be getting these similar effects because they're just symptoms. that are resultant of low oxygen that had nothing to do with the exposure to pHs. So how we select our fish is very thought out, but on top of that, we want to pick species that represent local species that in case, for example, something happened in the local watershed, we have an idea as to how sensitive they would be to those scenarios. So here's just a couple of these down here. And we ended up doing some of these based on the geographical location that we were trying to establish. And that was trying to find fishes that represent, across Canada. Not the easiest thing to do, but we picked, for example, your West Coast high ticket items, your salmonids, the fathead minnow for the central Canada and the western area that they're inhabiting tons of streams. Are very robust. They're saprinted. If something happens to them, we know we're in trouble. And then we've got yellow perch here, which is a fairly popular fish in this region as well. So this is actually, we got this from a local fish and wildlife club up in Kincardine. So pretty cool interaction with the community there. We also took all this work and put it on a gene chip and had a look at what that organism was doing at a molecular level within its body in the response to oil. Now, in two major scenarios, it was one, whether or not it had adaptations, or sorry, malformations or not. And that was a simple characterization if it looks all messed up or not, basically. But we wanted to find out what the molecular pathways associated with those changes were so that we can potentially go in and sample a couple of fishes and get an idea if they have been exposed to oil before, or if they haven't been exposed, are they susceptible to perishing, for example. And basically what we had done is, some pretty fancy colors, but we were comparing all these different mixtures of whether or not fish were exposed to oil, if they had malformations, if they're exposed to oil but didn't have malformations, but still were alive and functioning and fine, why? So those are the kind of signals that we were looking at. And a lot of this work is still going on because it's such an involved amount of research that we need to do in the lab, as well as do the actual oil exposures, et cetera. And this is kind of down the road of ecotoxicology. Now, this was all still pre-COVID, right? We got one more component to see, and then we started working on this picture came up, and I was thinking this is an oil sands processing area, right? And what I was thinking about more recently was the fact that this is the Athabasca River running right down here. Now, we know that the oil sands, is a major economic driver. It's just critically important, even especially now again. But we also know that its proximity to water and fresh water is becoming a priority in terms of how we're looking at water right now. And it's a bit of an issue when we're talking about things that you can see the sheens on these ponds here. And these are temporary berms that have been constructed in the settling ponds to put all the wastewater that has been used after it's extracted. Actually, it's used for extracting the bitumen, I should say. And so just the thinking of the proximity of some of these temporary berms and ponds next to the Athabasca River got me thinking about how I was doing things in oil research, and that maybe I need to expand to all the other components of the process as well. And that's where I ran into Diane Orahel, and she was looking at novel means of detoxifying the contaminants within that wastewater so that we can release this water back into the environment. New legislation has basically dictated that if you're going to use water for extracting oil sands, then when you return it to the environment, it has to be essentially background with nothing left in it that's toxic. And so one of the major components that's left as a result of this is these naphthenic acid components, these very acidic, very corrosive, but also very toxic compounds to fishes. And so we were charged with a bunch of engineers as well to figure out a method that they were using for naphthenic acid wastewater to see if we could actually detoxify it using novel photocatalysis. Now, what this involved is a really cool technology. It's fairly simple, but it's basically using buoyant microparticles that actually can be laid on top of a surface of water that react with the sunlight, that can produce reactive oxygen species that then degrade these naphthenic acids. So it's using the power of the sun to slowly degrade these things in situ. Now, as a result, we wanted to see if what happens after we actually use this process. Is the stuff in the water still toxic? And so we did our same suite again, where we took fathead minnow this time, looked at some an embryo toxicity bioassay, looked at mortality and malformations in this. Now, the hit zone of this one is the fact that we're looking at something that's in a natural environment. Instead of just going back to the lab and doing this all in the lab and figuring out, okay, these are our experiments, these are our concentrations, and these are giving us malformations. The next step of this was to actually do two exposures exactly the same way, but in two different environments. This one on the left here, for example, shows in an aqua lab down at Queens, where we're doing all these exposures in a very controlled environment. We've diluted these things down, we've treated them, we have the waters, everything's controlled, temperature, photo period, etc. And then my team and I went out into the field and did this in the field exactly the same way, and just didn't control for things like temperature, photo period, pressure, weather, things like that. And what we found basically was pretty cool. First of all, we know that the treatment worked. Basically, we have some organic compounds in the water from beforehand. We have our untreated naphthenic acid fraction component, which happens to be just a distilled water that comes out of the stuff that's been uh, extracted, so there's just naphthenic acids in it. And you've got a clear indication that there's other organic compounds in here. When we add this treatment and then use this photooxidative catalytic mechanism, you have a result in water that basically gives you nothing left in that. But because there's some these blips of other things that may be residual of what was being made, we had to check to see if that stuff was toxic or not. What we found basically is this worked really well. Not only did this whole process work really well, it actually cleaned up some of the background. So there was actually, in some cases, more fish healthier than there were in the control after a result of this high intensity treatment. And so just the process of moving this science into the field and trying it was a really cool experience. And you may think that a lot of us scientists do that, but bringing something directly from the lab itself and then replicating it in the field is extremely difficult, mainly because as you could probably see just given the pictures, these kinds of setups aren't easily movable. They're not overly robust. We've got microscopes, we've got expensive items, but at the end of the day, we're talking about fish and we're talking about whether or not they can survive as a result of this water being put back in the environment. So we make it work. Frankly, these little guys down here are little fathead minnows and they grew amazing. So there's actually still ongoing work on these exact fish as to what happened downstream, if you will. Then the anthropods hit. COVID-19 got right in the middle of all this, as it did for many people. And out of it, basically, a lot of things happened. First of all, the challenges. Now, for us as scientists, it was hard, especially in academia. The first things it was is the uncertainty. I mean, we didn't know if we were teaching or not. We didn't know if it was business as usual or if it was different. A lot of us had to change gears to remote teach. And we also had to kind of put things on the back burner. We still are dealing with the lag of what students are having to deal with in terms of academic requirements. And this will have a ripple effect later on. In research, basically, we've not lost, but we've impeded a whole training cohort in terms of what is our ability to get scientists out there in the field. It's not easy to do that virtually, but we'll talk about that in a second. Even things like access to laboratories has been difficult for us. Shipping has been a nightmare in a lot of cases. And clearly, in a lot of cases, you'd probably recognize that PPE is hard for us to get as well. And some of our work requires it as well, and it's just not a priority given the scenario. But also things like access to field work and how we do our collaborations out in the field were challenged. A lot of us who do theory work, if you can get into the lab, that's one thing. If you do applied work, you may be able to do a field season with, the social distance restrictions and things like that. But it really started to change how we thought about things and collaboratively and integration of themes and whatnot. And so a lot of us started to think about how to change our focus. A lot of us also started to think about how we're communicating our work. For example, I think at this point, even right now, conferences and Zoom are not the same. There's an interaction component that happens with being in the same room with someone for a while. You get familiarized. You don't get that same kind of panicky, conversation that you get going on. You also have a chance to have a beer or so and, work some things out, meet some old friends, if you will. We are trying to get around these things with stuff like breakout rooms, et cetera, but they're just not that social. They don't have the same kind of opportunities a lot of this has. But on that same notion, we can say that a lot of these things can be flipped into opportunities. For example, this particular word, not in my backyard, yeah, it has a pretty negative connotation, but it also means that people are paying attention to things that are in their backyard. So if we can flip that into a good thing rather than a bad thing, that's one thing. But basically, this leads to a reappreciation of the surroundings, as I say. People are starting to move into these smaller towns again. They're starting to come from rural areas. It's this boost in resurgence, trying to get out of the cities, if you will. This also leads to really improved connectiveness, as we're finding out right now through virtual aspects. But it also leads to an awareness environment. With more people spreading out into rural areas, people are paying attention to what's actually in their backyard. And maybe not in my backyard means I'm going to do something about it in a good way, not in a negative way. This also corresponds to something that we're doing right now. We have an audience and we have people paying attention. So this is the first step in communicating. I'm going to hit on this part a lot coming up, and again, not very much longer, but the community component is huge, and you're all very important in that. The idea is that we can now integrate your interests, even help getting people training and do some citizen science on this is a huge component that was a benefit that maybe we wouldn't have had that push to get to without COVID. And the good and the bad. A lot of us are interconnected way more than we used to be. And for science, this has exploded. In fact, I don't know anyone who's in science who isn't working 4 times harder now than they were before COVID, based on how connected we actually are. But at the end of the day, this is what we're looking for. This could be an actual new chance to reconnect the whole of science and community together through our environment. So I offer something that I've been thinking about as a physiologist, and physiology works on feedback. And when I think about this post-COVID science scenario, it's pretty clear that we've got a new era ahead of us, and maybe working together is a good time to start these kind of relationships immediately. We know that the new perspectives from people that are listening, especially to things like this right now, really facilitate how we move forward together as a community, as well as a scientific community. An important component, especially these days, it's also a voice for what matters to communities. To get involved, to say, there's problems in my backyard. The lake is messy. The river's all over the place, right? Like we got the river levels are up and down. How do I do stuff about it? Those kinds of that kind of dialogue is important to actually get stuff done about this. And as I mentioned before, one of the aces in the pocket, if you will, is citizen science. Back in the day, it used to be just clipboards and pencils, but now there's phones and videos, and we're seeing all this kind of stuff. Even some Reddit subs have some amazing components that I use in my teaching because there's people out there in the field seeing this stuff and recording it. Oops, going backwards. The communicating part is obviously important as well. Getting our results out there to show what we're doing is important, and you can see that evidenced in some things like the fish advisories out there now for fishing regulations. Things are interactive maps, so people can actually interact with what their particular locations are. And on top of that, feedback to those components to say, hey, you're missing areas here, or we need to know more about this particular area. Why is there no data on this? And this allows us that dialogue, if you will. So overall, I've been trying to pull my research back to the environment based on what has been changing as a result of COVID. Things are rapidly changing. Right now, we just had 34 here in Kingston yesterday. Now it's like it was 15. And this stuff's happening quickly to us. Now that's climate, I get that. But basically, I've been thinking is how can I use my experience to bolster the scientific integration into new areas of environmental research? And that's where I got into working with the River Institute. Now, before I got the whole perspective of the River Institute, I understood that there's some people at Queen's also doing similar components. Now, in this scenario here, I decided to think about what kind of future environmental challenges are we running into in this area? Yes, eutrophication, obviously. Those are water-based issues still. Eutrophication of waterways and the bioremediation of chabs. So chabs are cyanobacteria, harmful algal blooms. These are very important components that are coming out these days that we're all experiencing. I mean, the advent of eutrophication and phosphorus loadings in the nearshore areas and in rivers and smaller lakes and ponds are leading to these massive algal blooms to the point where things are getting fairly dangerous fairly quickly. So I decided to join a group at Queen's here, Dr. Lefebvre and Dr. Wong again, as well as some other components that are from our Chinese collaborators. And they've been working on this snail that actually is really good at eating some of this stuff. And, this is all ongoing work, but basically we're trying to find out whether or not the local snails and Bellamia snails in this area are able to be used as bioremediation methods. They're actually filter feeders, but they don't actually necessarily have to eat the algae in this case. This particular guy here, the Sanotia aeruginosa, is known for producing 2 different types of poop. One is pseudofeces and the other is feces. And what these are this particular pictures, are microcystin, or the cyanobacteria, containing feces. And instead of actually digesting the stuff and removing it from the water, what it actually does is wrap it up in these coatings, these vacuoles, if you will, and it just delays them from congregating and making algal blooms. And so really what we're talking about is delaying or preventing blooming events rather than cleaning them up after they happen. So it's a proactive solution. A lot of this stuff is being driven by a really special guy named Alan Tian in Queens. He's also involved in UAV monitoring of some of these things and have developed particular algorithms to actually understand how algal blooms are being created, under what conditions, and how they're moving around. And if you see him out there in your local area, he's usually out there with a drone and water sampling stuff, looking for all sorts of fun things. Another component that he's looking at as well is trying to add a survey aspect of what's out there. Now, using eDNA, which is environmental DNA techniques, and that's basically any kind of messages that are released from fishes or anything that's living, essentially, that you can essentially find out in that watershed what's there, what's present. And so as a survey technique for things like invasive species or for algal blooms that are, that have microsystems in them, it's a really good process to use to go out there and see what's out there before problems arise. And he's been running that for a few years now. He just started his PhD, and you'll see him around the area with his drone up and taking water samples. Pretty cool stuff. The community component was really important. Alan really made me think about his work. He's in people's backyards, talking to people, understanding what they're seeing for eutrophication, how they can identify algal blooms that are harmful versus not harmful. And basically, he inspired me to think a lot more about my involvement with the community. In fact, how I could integrate my research in that was my main thrust point. And so just coming back around to the River Institute again, is I met a bunch of special people there and a bunch of special projects they were working on. And they've been very inspiring. And I think I'm just going to give you my take on why I think these are important. So the first is Matt Wendell's here. And again, these are by no means an exclusive list. These are just some of the things going on that I've been involved with. The River Institute does a lot of things, and I wish I could be part of more of them, that's for sure. But Matt Wendell's particular fin survey that you may see him going around with this large team of volunteers collecting fish everywhere. I mean, look at these numbers: 205 sites visited and 130,000 fish sampled. I know how much work that is firsthand. And it's a very exciting project. And I'm really excited to be working with him and his group and looking at some of these eDNA methods, as well as with the Queen's group, for looking at surveying some of those species that I was talking about, whether or not the invasive species like tube-nosed goby and tench are migrating their way down the watersheds or not, especially in the north of Kingston where Alan's looking, or where Matt's looking in the St. Lawrence itself. We also know that these are great methods for looking at species at risk, whether or not if we find messages or cellular messages, eDNA, that they were actually there in that region, it allows us to target our conservation efforts much better. Now, this is still in development, but you can see there's a large ticket item fish in this one, for example, American eel and lake sturgeon, that are very high level priorities that can be bolstered in terms of helping their conservation strategies with these kinds of methods. Another one is just showing, really, driven by Louis Savard here, the River Labs program leader, is a multispectral assessment of how phosphorus moves across the landscape and gets into a watershed. Now, it's not quite the same as the St. Lawrence River, but it's Lake Simcoe. And what the demonstration of this project is the fact that the River Institute is reaching outside of its zone, if you will. They're doing things so well that people have caught that on the wind, if you will, and now they're getting to help them out doing other projects. And this particular project is neat because it shows how drainage in an area leads to eutrophication in a particular watershed, that then we can target our approach to look for things like cyanobacteria, harmful algal blooms. So instead of wasting our time looking around the whole lake to find these areas that we should be concentrating our efforts on to find the sources of these eutrophications, we can basically use modeling to figure out ahead of time where we should be focusing our efforts. And this is another one of those larger collaborative projects that is just really inspiring, because it involves the local Lake Simcoe and homeowners association up there, with all these people whose backyards we're actually in, who can help us make these links as to, do you see problems? Where do we go first? Can you find people to help us? Can we communicate our results to you? Can you send us back feedback on what we should be looking at? We're not living in there, so can you help us? This kind of interaction, this relationship, is extremely important when you're not able to be on site all the time. And, it'd be ideal for us to be there all the time, but it comes down to resources, and the community is huge in driving these resources in the future. The last component that I want to touch on is a really, really special project. And I think many of you out there know how special of a project this is. I think it's one of those things that kind of caught me off guard at first. I wasn't quite understanding exactly the mechanism to what was going on, but after spending several months with this group, working with them, it's amazing. I mean, honestly, the Great River Report itself is such a unique take on things that I've been very inspired by it, I'll be honest. I mean, to look at the ecosystem health as a whole and not just from one set of eyes or perspectives really makes sense to me. And in fact, I'm getting goosebumps thinking about it. It really it hit home with me about why the community effort is important. It's tying everyone in to say, look, it's all of us, and it's as perfectly down here how the health of the ecosystem is intrinsically linked to all of us. Now, this is an important component, especially someone that's done, very intense lab work and feedback and all that kind of stuff. This is real. This stuff is important. And I'm really happy to be part of this to at least help out with some of the contaminant work on this, given my expertise. And we're looking at some of these components we're building into this report and the fact that it'll be released shortly, hopefully. But I just want to say, looking forward in terms of how this report is being pulled together, I know there's tons of people involved in this, so you should all be congratulated for how it's going. But basically, looking on how we go forward in this in science, I look at this as an archetype, especially as a shining example of how we come out of these dark times with COVID by involving more people rather than fewer people, right? The last component that I'm working on isn't directly with the River Institute, but it's a really special project. Clearly, most people at this point probably know Abraham Francis, who's a shining star in his own right for sure. He's been very inspirational to me, especially for, I first met him and he was chatting about the meaning of the river to him and his family. It really touched my heart the same way the lake where I'm from had the same kind of meaning. So we're working on this really cool project right now between myself, University of Gleer with Ken Druard, and Satindra Bhavsar of Ministry of Environment Canada Parks or Conservation Parks. And basically, we're looking at updating the fish consumption advice around the MCA area or the Cornwall AOC region to include a more balanced, less prescriptive risk-based approach and a more balanced risk, benefit, and perspective approach. So a lot of this work will basically involve linking the benefits of fish consumption as well as the risks, your typical contaminants and your loadings and things like that, to get a better comprehensive perspective as to how we should move forward with advice for consuming fish with respect to all cultures. Now, at the end of the day, this kind of informs my research moving forward. I've got this whole circle of experience, for example, that we've gone through, but one thing is different. And the thing is after learning all this and having all this experience in these different facets, something has changed significantly. And unfortunately for me, it's a denominator. It changes everything again, because everything that I've spent time looking at is in the context of a certain temporal aspect, that we have patterns and cyclical changes and environmental cues, and that these are all coordinated efforts. Well, what happens when you just turn the speed up on that a little bit? Well, you have to kind of go through this whole process again to understand how an animal is interacting with its environment. And so to me, this is kind of represents a sea change in terms of how we do work in the future. But to leave you with one last component of it, basically, what has the community taught me about my own research? Well, I can tell you right now, it has been one hell of a journey given the number of students I've been allowed to be part of their careers and the work that they've done. Here's Jesse, one of my favorites here as well. The quality of students and mentorship has been very inspirational to see what they go on and go do. Now, remember, it's not just science that I'm hoping that they go do. I'm hoping that also train new scientists, critical thinkers, and community leaders to be part of that. We need everybody involved in this, and building these integrative networks is extremely important to push the narrative of what science actually is forward. We all own it. It's not one of us, for example. I also also recognize that there are a number of challenges to academic research and education out there, and access is by far the number one, especially coming from a very small town, not a lot of money. That's a tough one, and I personally understand that, and I will do my best to help correct that. I know also firsthand that diversity, different ways of learning, different perspectives are all huge strengths in science, and I cannot state that enough. In fact, I've had many examples of that in the last few years that has shown me that that's the only way to go. Also, it's really important to find those who can ignite the spark and cultivate passion. And this is being part of a team and not necessarily always being an individual in that case. And one of the most important things I've learned in this journey, especially during the anthropause, is the self-reflection and the flexibility of my work, being able to roll with the punches, as it were. It's really important, I think, right now, especially in this time, that we help redefine our relationships between us, the environment, science itself as a process, and the community. Because basically, we need to reestablish some of these relationships between us and among our environment and our brethren as well. And a lot of this comes down to building dialogue and getting feedback. And that a lot of that feedback component, I think most people, at least myself, have overlooked over the years. And one of the largest components of this feedback network is just to listen. And with that, I'm going to end the talk. Oh, one last thing. Sorry. Had to.
Thank you, Barry. Thank you so much for taking the time. And I mean, just to see you condense so much of your research actually in a very short period of time. So thank you for sharing that. And we're just a little bit late on time. So I'm just going to say bye to everybody who's watching from your TV. And thank you again for tuning in. In August, we are taking a break from Science and Nature, so we will not have a talk for that month, but we'll be back in September. And for now, if ever you are interested in some workshops, you can visit riverreport.ca/workshops, and there may be something you find interesting. Have a wonderful evening. Great. So thank you, Barry. I'll open up for discussion. I do not have any questions yet, but I imagine they may flow in. We got a hello from Philip Ling from Maitland Tower. He's a very big supporter of the River Institute and all the research that we're doing. I think you're muted.
Yeah, sorry. Yeah, no, I said absolutely and said hi.
Thank you.
Yeah, I realize there's a lot to go on, but as I said, there's many aspects to that I can speak on. I think it's interesting just to, more recent events have been very eye-opening as to what I had initially set out on my career, for sure.
I know that it was particularly difficult for anyone working in academia or in the ministries to do research this last couple of years, or a year and a half, I guess.
Yeah, I mean, I'm not suggesting that, as an academic, I'm crying about it. I'm suggesting that it's been a challenge for all of us. But to be honest, the challenge in academia comes from us, a number one priority is the students, and having to support that at the right time in the right way, not knowing where we're going right or left kind of thing. And that's the challenge is, you have to understand it's been 2 years where people in a lot of cases haven't even had exposure to practical lab skills, which is what we are. It's part of our process, right? So that's a tough one when you're talking about skills that you have to work on and continue to keep up. It's a challenge for a lot of people.
That's what happened for my ecotox class with Jeff Reidel with the environmental tech program. Yeah. We had to do everything theoretically, and on paper we couldn't actually do the tests in the lab. So,
well, the funny thing about that is that I have pipetted thousands and thousands of times, and I would still have to go back and make sure I do it again properly, because it's a learned skill. And if as soon as you stop doing it, I used to skateboard as a kid, and I can't do that anymore, for example.
I guess, like, speaking of about pipetting, you were just mentioning the eDNA for citizen scientists. I know it wouldn't necessarily be pipetting, but would you be able to speak a little bit more about that?
Absolutely. That's an excellent part. So eDNA is an interesting thing because it's a very specific technique that is really just dependent on how you go ahead and do your prep for it. At the end of the day, you're just getting a water sample, and then you're going to filter that. And so for a citizen scientist kit, in fact, without getting into the details of it in that time, but Alan Tian and I have been developing a low-cost citizen science eDNA sampling kit, just so we can get something out there to mail out to the people that are interested, and they can take water samples. All you have to do is take like a 1 liter water sample, filter it through a certain membrane, and then that membrane's got the stuff on it. So you can send us that membrane, or we can come pick it up, or we can come deposit into a local area if you got a water authority body that can collect them for you or whatever, and then we can process it. But the thing is, it's a water sample, so it's nothing specific until you look for a particular message. So all of the work comes on the lab side. So for a test kit, it's actually pretty feasible to send something out there to have someone in the community take a quick water sample of, say, that's a suspicious algal bloom or something like that, filter it, and then send it back to us in the mail, and we'll be like, oh, that's a bad one, we'll put it on our list, and we'll put a little interactive dot on our map, and you can, feedback with that. So
oh, sorry, I just saw there's a message that came in. Yeah, I guess to come back to that, I think that's a really interesting kind of activity for citizen scientists, and especially for this, like those who are doing the tests, because then it's not kind of going into the dark. It's like you already have, like you said, you already have these pins on the map of where you could check out. And
yeah, so like I'm glad you said that, because I'm trying to wrestle with even building this into labs and stuff that I teach. And I think the big issue is you still need a little bit of infrastructure. You still need some molecular stuff. That is where all the money's spent. But to get I should have prefaced this better. The more data, the better. But the problem is you need to be able to put that data in context. And if you're just getting data for data's sake, that's where we are right now. But if you're getting a large amount of data that's already been vetted in terms of how you collect it, it's going to be verified, i.e. you build those relationships within the community efforts before you actually run these papers, and you end up getting really good people getting really good data that you can't possibly get yourself. And if you can get that feedback network together, well, you're laughing, because half of your cost is getting all your team out, writing the forms. And if people are sending back and they're all eyes and ears, like that's a huge component. Now, I do say that with some caution, because the caveat is, bias is a real thing for everybody, including scientists, even though I don't think they have it. In fact, we probably have more of it. But being aware of your bias is all you need to have, right? So what I'm getting at is, if someone sees something off of a dock, they're more likely to look for that as a problem. And so we understand there's a data issue, and that's where it's a training component. But the physical act of going to get something that's a sample that we can then analyze later, to have thousands of people doing that across a nation would be the ideal scenario. And that's how science should happen, if you ask me.
yeah, I'm excited. I look forward to that. I imagine, and let's say when this does end up happening and is available, how will the community know about it? How will we?
We'll have to get off our asses and do it. And that is, I'm going to have to get out there to the community and talk to people and get some people together and train them. And so, again, I want to be very clear about this. This is not a flash in the pan solution. This is something that we have to build that rapport, so that the community understands what we're trying to do. We understand that we're trying to do for the community. We're clear on what those links are. And that if we build that feedback network that works, it'll work well. But it has to have, the vetting process, the trust, the communication has to be ongoing. And to be honest, at the end of the day, the community needs to understand that we are working for them and with them, and not against them. And maybe we have to do a better job as scientists communicating that.
Great. Well, thank you, Barry, for speaking to that. I did get a question from Phil. He is asking, so are there any citizen science tools available to evaluate stress in fish in the field rather than in the lab?
Well, that's a very interesting question, because it's a very tough thing. So, okay, let me just preface this, back up for a second. Stress itself is by definition a challenge to homeostasis. So I got to ask you, how would you assume that there's a challenge to homeostasis in an animal? That's really hard. And so what people have tried to do in historical times, at least, is add proximity measurements that would indicate something. One of the things that I think a lot of people are aware of is plasma glucose. If you have a stress response, a reaction to something, allergic reaction, or you're hypoglycemic, hyperglycemic, you have a lot of glucose, but you have no idea how you got there. As a human, you can describe that, but as a fish, they typically would deal a lot of times with higher glucose levels. So that doesn't work. It's not a good indicator. So all we really have are the hormones that are associated with stress, and that is like cortisol in this case. But we're talking thousands of dollars to run like cortisol assays to do this. So there's no easy way of determining stress. However, now that I've said that, I have some information that I have to be very careful about what I release, but I know there are people working on non-lethal methods of determining stress based on proteomics and things that are available in stuff like mucus. So if you can get a mucus scraping, you might be able to discern some kind of transcriptic or genomic or proteomic, I should say, sorry, profile. And if that profile can be vetted ahead of time through a series of laboratory experiments, you could say, oh, this fish is in category B, it's stressed, for example. That could be a reality. We're not there yet.
Great. Thank you, Phil, or Phil. Thank you,
No, thank
Phil,
you,
for
Phil.
your question.
That was a great question. No, that's a challenge for the ages, Phil, and we're slowly getting there. And the way things with quantum dots and all the technology is we're really getting close. We're really getting close to that. The problem with hormones and endocrinology is that you need such tiny amounts. So what you can measure at the limits of detection are obviously still having effects in some cases. So that's part and parcel of endocrinology, unfortunately.
Well, thank you, Barry, for answering the question. I have a comment. So this is from Lee. She says, excuse me, not a question, just a comment. Thank you, Barry, for sharing all your work. Just the scale of your work from atoms to whole systems is such an amazing breadth to cover. So important to have people in science who are able to see the whole picture.
I thank you for that, Lee. I mean, to be honest, there's 2 points of that. One is I've been trained by some very amazing people, very privileged to have been done so. I also had been working with some fantastic students, and I'm talking like life-changing, fantastic students, and I hope I can continue to do so. And I'll flip that back around on you and say the sheer magnitude of the stuff that you're doing on the river report, for example, blows my mind too. So right back at you.
Thank you, Barry. It was very nice at the end to see the projects from the River Institute, and it also made me realize just like the breadth of work that we're doing and just recovering so much.
There's so much going on there that I had no idea until I lifted the hood, and then I was like, I'm not a mechanic. No. Honestly, all I touched on were the projects that I'm personally involved in. There's so much going on and so many other components that I'm so impressed with. Again, I'm not fawning over it. All I'm suggesting is that I'm extremely impressed with the River Institute's ability to incorporate the community directly into the work. I have never seen that anywhere else. And I really hope to promote that, as well as maybe co-opt some of that when I move out of this area and start doing it somewhere else. But that is the scenario that is, I've really had my eyes glued wide open because of what you guys do down the River Institute. And you guys should be commended for it. It's really, really special.
Thank you so much. We should have you as our spokesperson.
I'm a scientist. You don't want to hear from me. No, I'm just stuck in my Excel sheets or something. But yeah, no, it's been really inspiring. As I said, it's lifted me out of this. From when you're in a lab and you're doing very specific work, you get tunnel vision a lot of the times. And you can tell some of the work I've done is very esoteric. But then you step back and realize how you can help some other projects based on that knowledge, and it's oh wow, it's synergy, and that's what science should be, right?
I totally agree with you. So I guess I mean I'm not seeing any more questions.
Oh, no. It's like my classes. I talk too much stuff too quickly. My apologies, but I'm always available to reach out and chat with. That's for sure.
Exactly, and these presentations are recorded, so you'll be able to share it as well. We'll be able to reshare it. So it is the middle of summer, and I imagine a lot of people would rather go out and have a beer or be outside. So
I gotta get back to those projects, but you can also watch it on 0.75 speed. And I hear that's what a lot of my students do. So
I'm not familiar with it.
Oh, just instead of watching everything at 1.5 speed.
Oh yeah, okay, yeah.
When it's me, they watch it in slow motion instead, so that's fair. No, this is a great forum for people to communicate. As I said, I wish if it were a different scenario where we had more time, that's another thing. But these are special events for sure.
Well, thank you for being part of it. Yeah, and on that, I guess I'll say goodbye to all of our viewers from Facebook and Zoom. Thanks again for tuning in. Thank you, Barry, for taking the time to come in and to create the presentation for us. And okay, sorry, I saw there was a message that popped up. Okay. And yes, so we do have those workshops that I highly recommend for everyone to watch, to join. So those are riverreport.ca/workshops. Great. So thank you, Barry. Thank you, everybody. Have a good night.
Thank you.